DNA footprint assay. It is a technique that helps scientists discover the location where a protein binds with the DNA. In the first step, we amplify the DNA. For this purpose, we take a specific part of DNA that might have protein binding sites in it. Now we amplify this DNA using a technique called PCR. In the next step, we label these amplified DNAs so we can track them later. We often use radioactive labels, but other methods are possible too. Now we mix the labeled DNA pieces with proteins. For comparison, we must take another sample that contains labeled DNAs but lacks proteins. This sample is generally known as the control sample. Proteins in the original sample bind with the DNA molecules at their binding sites. Next, we add an enzyme called DNase 1 into both samples. This enzyme cleaves DNA molecules at relatively random positions. Notice, in the original sample, this enzyme can't cleave the DNA where protein is attached. Finally, we run both samples on a gel in gel electrophoresis. The DNA fragments get separated on the gel based on their size. We use a detection method like X-ray film to reveal the band pattern. Notice that the control sample shows a complete ladder of bands, while the original sample has gaps. These gaps are footprints of proteins on DNA molecules. They depict the position where the protein was bound to the DNA. The main purpose of DNA footprint assay is to study gene regulation. Proteins that bind to DNA can either turn genes on or off. This way, they influence the cell activity. Scientists can understand the regulation of genes by discovering these protein binding sites.